Hi everyone, my name is Maria, and today I'm going to talk about long-term survival and the possibility of life elsewhere from the perspective of halophiles. Halophiles is a general term that we use to refer to microbes living at high salt concentration. Salinibacter ruber is an interesting halophilic bacterium. Its physiology resembled the one of Haloarchaea, and that's because throughout its evolution stole some genes in order to survive in hypersaline environments. Dunaniella salina is a green microalgae, an eukaryote that is responsible of the primary production in hypersaline environments. And then we have the main character of today's talk, an archaeon called Halobacterium salinarum, able to survive up to 5.2 molar of salt, and it has a striking features that makes it a model organism to investigate the limits of life at two extremes, salt and time. These halophiles share the same environment. However, these environments are really dynamic and subjected to period of desiccation, in which water evaporates and salt crystals precipitate. Inside those crystals, little pockets of brine remain encased within the crystal lattice, and halophiles within it. For some of them, this is too extreme, and they die off, like the cells of Dunaniella salina that lyses and releases intracellular molecules. But Haloarchaea, like Halobacterium salinarum, are able to survive. But the question is, how long can they survive inside those little pockets of brine in the crystal lattice of salt? Well, there are many studies that have found not only genetic material, but also living cells inside old and ancient crystals of salt. This has remarkable implications concerning the resilience of these organisms, the physiological mechanism behind their survival, and the strategies to preserve DNA and cellular structure over geological timescales. Now, different hypotheses have been suggested, like the recycling of molecules coming from dead organic matter, or an innate ability of the specialists in long-term survival that are not fully understood yet. Fluid inclusion is the technical name that we give to these pockets of brines encased within the salt crystal. The aim of my study is to understand how can Haloarchaea survive over geological timescales in a closed system like the fluid inclusion. And to do that, I'm testing two hypotheses. The first one is the existence of alternative metabolic pathways, and the second one is the death of part of Haloarchaea needed in order to have a pool of carbon and energy sources to sustain the metabolism of the surviving ones. However, there are some constraints. The first one is we have to constrain the timescale that necromass recycling can maintain or sustain their metabolism. But there are also stringent thermodynamic limitations due to the amount of energy that can be gained. And the last one is that necromass recycling over long time scales is limited in a closed system like the fluid inclusions. There are three things that I'm also taking into account. The first one is constraining the energy gain and energy limitation of alternative metabolic pathways. The second is the dwarfism, a phenomenon that may be triggered by starvation, that will decrease the cell volume and therefore decreasing the amount of nutrients required to sustain the metabolism. Also, changes in the proteome without relying on the synthesis of new proteins that costs energy. The first objective is to get insight into mechanisms allowing Halobacterium salinarum to adapt to fluid inclusions through proteomics. And the second objective is to determine the survival of Halobacterium salinarum in laboratory simulated fluid inclusions. In the first objective, what we did was growing cells of Halobacterium salinarum in normal growth conditions. And once they reached the late exponential phase, we collected the cells, we washed them, and we resuspended them in a salt solution in order to make sure that all the nutrients were removed. Then we poured the solution with salt and microbes in big petri dishes and waited until the crystallization was completed. That represented our second time point. We also added a third time point that is represented by 42 days inside the salt crystals. But when we compared the first two time points, what we found was that many enzymes involved in the translation process were significantly downregulated. We also found that an enzyme involved in the arginine deaminase pathway was significantly upregulated, and the arginine deaminase pathway is an alternative way that Halobacterium salinarum has in order to produce energy. We also found that the dimethyl sulfoxide reductase, an enzyme involved in the anaerobic metabolism, was significantly upregulated. And this makes sense because what happens in the fluid inclusion is that the oxygen gets consumed, and therefore the microbes need to switch to an anaerobic metabolism. The biggest changes that we found were when we compared the proteins extracted from the first time point with the third time point. We found that 79 proteins were significantly differentially expressed, and we found that all the enzymes involved in the arginine deaminase pathway were significantly upregulated. And interestingly, the tRNA ligase of arginine was significantly downregulated, suggesting the idea that the microbe is preserving the arginine, and instead of using it to produce new proteins, is using it for the arginine deaminase pathway, that is an alternative way that the microbe has to produce energy. We found that many enzymes involved in the translation and in the transcription were significantly downregulated, and the dimethyl sulfoxide reductase was significantly upregulated, supporting the idea that the microbe is switching to the anaerobic metabolism. In a paper published by Sorokin et al., they suggest the idea that a single enzyme of the DMSO reductase family is responsible for various sulfoxide-dependent respiration in haloarchaea. So the question that we are asking now is, can they grow on methionine sulfoxide and tetramethylene sulfoxide? The fact that many enzymes involved in the translation and in transcription were downregulated, significantly downregulated, suggests the idea that the microbe is trying to preserve the energy in order to modify its proteome instead of producing new proteins. However, among the many enzymes that were downregulated involved in the process of translation, we found that one single protein was significantly upregulated. And now we are asking whether this protein is responsible for the silencing of specific genes. In the second objective, the aim is to model the survival inside the fluid inclusions. And what we did was, again, growing the cells of Halobacterium salinarum until the late exponential phase was reached. Then we collected the cells, we washed them to make sure that all the nutrients were eliminated, and then we resuspended them in salt solution. But this time, what we did was to simulate a big fluid inclusions by pouring the salt solutions with microbes in it in glass tubes and sealed them anaerobically in order to mimic what happens in the natural fluid inclusions. So in this second objective, the entire volume of cells inside salt solution were divided into glass tubes, each glass tube representing a single fluid inclusion. Now in this graph, each dot represents a single glass tube that was selected, the OD was measured, and then a serial dilution followed in order to see how many cells were still alive. And as you can see from this graph, over a period of 144 days, what happens is that initially we have a decrease in the population, in the survival, and then they reach a steady state. And we think that the first decrease in the survival of the cells is needed in order to have a pool of dead organic matter in order to sustain the cells that are surviving. We are now asking whether this is an active response of the cells of Halobacterium salinarum, or is just a random event in which cells are just randomly dying, and then cells that are surviving are taking advantages of the cells that died by recycling the nutrients of the dead organic matter. So, in the first objective, we found that the vast majority of the enzymes involved in the translation and in the transcription were significantly downregulated. And there is evidence of strategies to long-term survival with the upregulation of enzymes involved in alternative metabolic pathways. In the second objective, we found that initially cells of Halobacterium salinarum decrease and then they reach a steady state. And we think that this is needed in order to guarantee the survival of the other cells that are surviving. Now, the future direction of these two experiments is to, first of all, continue with the time point analysis, and second, look into the phenomenon of dwarfism, both visually and biologically through proteomics. The ability of Halobacterium species to survive over geological timescales with little or no nutrients makes them interesting model organisms to investigate signs of past or present life elsewhere. But what about radiation? So I decided to expose the crystals to ionizing and UV radiation, high doses of these two sources of radiation, and also with different setups of halophiles inside the crystals. And as you can see from this graph, Halobacterium species were able to survive with different lag phase, and lag phase is represented by the height of these bars, and the bigger they are, the more time it took them to recover after the radiation. This experiment is still a work in progress. There are new results that are under analysis in the lab, so I hope that for the future I'll be able to address more specific questions related to the radiation. And with this, I would like to thank you all for the attention. I'd like to thank my supervisor, Terry McGonigle, my lab team, and my funding body, the Soul Giant. I'm happy to answer all your questions. Thank you very much.